All right. Well, without further ado, let's get this webinar started. So once again, thank you everyone from us here at Elixirgen, as well as our colleagues over at Ananda Devices, to everyone who has joined today for today's live webinar of iPSCs and Microfluidics, Innovative Approaches to Modeling Alzheimer's Disease. Our speakers today are Dr. Michal Millrod from Elixirgen Scientific, and the CEO and co-founder of Ananda Devices, Dr. Margaret Magdesian. Uh, just a little breakdown on today's webinar. We will be letting both speakers go through their presentations, and we'll have a full Q&A at the end, as well as for those who joined the last webinar, we'll actually be adding an additional discussion session near the end of the presentation with some really pretty exciting topics. But just to help with the presentation portion, we will be having the Q&A at the end. So if you do have any questions, please submit them through the comment section. And at any point during the webinar, the only thing that we would request is if you have questions in regards to a particular slide, to please use the slide number as well as the presenter's name, so we know which slide to pull up to help answer your question. If you would like to send a question privately, please use the email questions@elixirgensci.com. We'll be able to pick those in and be able to answer. And once again, if we don't get to everyone's questions, please know that we will follow up with you in regards to answering those questions afterwards via email. So please make sure that the email that you have in there is the accurate one that you use to register. And without further ado, I think we can move on to our first speaker, Dr. Michal Millrod. She got her Ph.D. in immunology from the Molecular Biology and Genomics program at the Johns Hopkins University School of Medicine, where she also took the next step going through her postdoc as well. She joined here at Elixirgen Scientific in 2020 and has over a decade of experience culturing and differentiating human pluripotent stem cells. She specializes in creating consistent, intuitive, and easy to follow user guides to ultimately save clients time and increase their efficiency so they can focus on achieving their research goals. Ladies and gentlemen, Dr. Michal Millrod.
Hi, thank you so much, Henry, for the introduction. For those of you who have attended our webinars before, hopefully you're already familiar with Elixirgen Scientific, and for those of you who are new, welcome. So just to briefly introduce you to Elixirgen Scientific, we were founded in October of 2016 and have locations in both Baltimore, Maryland, as well as in Japan. And our company is really centered around what we call the quick tissue technology, which is essentially a transcription factor-based differentiation method that allows us to efficiently and rapidly differentiate iPSC to unique cell types. This quick tissue technology relies on the use of a human gene expression correlation matrix, which was developed by our founder, and we use to identify genes that are highly correlated to the specific cell type of interest. And we then produce a cocktail of synthetic transcription factors based on the gene correlation matrix results and test them out to figure out the ideal one for the cell type of interest, and then optimize the differentiation conditions, media, matrices, etc., for each individual cell type. Our differentiation technique allows us to have several advantages that are not found with other differentiation systems or with other protocols that are based on growth factors or media, but including it being a very systematic method. The differentiation is rapid, it's robust enough to work with multiple different iPSC lines. The cells produced are highly functional and they do not have genetic integration. There's no genetic footprint because we are using mRNA technology. There's not a viral integration. The process is very easy to follow. We give great user guides and customer service and have a very similar protocol for different cell types. So once you use our cells and are familiar, you can easily use other cell types as well. Using our quick tissue technology, we've developed a broad portfolio of products and services. We have differentiation kits that allow you to perform the differentiations in your lab. We sell frozen cells that we've differentiated here. All of this can be done with different iPSC lines. We have access to the CERM lines, but we can also help facilitate procurement of various iPSC lines to meet your specific needs. And the services branch really customizes based on your project and your needs. Some examples of some recent services have included differentiating cells from different patient-derived lines, as well as cell lines that are provided to us by a particular customer who might be working on a specific mutation or disease. And we differentiate the cells, and we can perform various downstream assays and give a complete report at the end. So again, we encourage you to please reach out and let us help you with your research goals and provide a customized service for your needs. Today in particular, I wanted to illustrate how our technology and cells can be utilized to generate disease models with relevant phenotypic expression for discovery and development of pharmacological and other therapeutic approaches. And in particular, in honor of World Alzheimer's Month, we're going to focus on Alzheimer's disease, which is one of the most prevalent neurodegenerative disorders, and it's characterized by a progressive lack of memory and cognitive function. And yet, despite decades of research, our knowledge of the disease is still quite limited, and a huge impediment has been the lack of suitable models. Animal models have fallen short and are not always translatable downstream to the human studies. And using iPSC-based models allows us to create patient-specific cell lines and truly study the disease. And a big impediment to iPSC-based models has been its requirement of a highly skilled researchers who are familiar with it, as well as the cost. And by allowing Elixirgen to help you, we can surmount some of those obstacles. So through our license with CERM, again, we have access to a lot of human patient-derived iPS lines. And again, today we're focusing on Alzheimer's disease, but feel free to reach out if you have other disease interests. We have lines for them as well. So again, our technology allows researchers to obtain standardized, commercially available neurons. And in this case, excitatory neurons were generated from age-matched healthy controls and an Alzheimer's patient. In this case, they were an ApoE4 carrier. And you can see the typical morphology of the neurons with neurite outgrowth. And we looked at the expression of neuronal markers, such as the pan-neuronal marker tubulin beta 3, class 3, as well as the glutamatergic neuron marker vesicular glutamate transporter 1. And this is at day 10 of differentiation, which would be day 7 of post-thaw if you buy our frozen cells. And these were actually maintained in culture for 6 weeks. And you can see here that the neurons are able to differentiate and function and express the relevant markers. So some of the main features of Alzheimer's disease are abnormal accumulation and aggregation of amyloid beta protein plaques, as well as tau tangles. And therefore, these two proteins are used as biomarkers for disease detection, as well as targets for drug discovery. So amyloid beta comes as amyloid beta 42, or there can be amyloid beta 40. 42 is a 42 amino acid peptide that comes from the cleavage of amyloid precursor protein, and it aggregates and forms amyloid plaques, which are one of the hallmarks of Alzheimer's disease. So in early stages of Alzheimer's, the amyloid beta 42 in the cerebral spinal fluid actually decreases because it becomes sequestered in these brain plaques. Amyloid beta 40 is also derived from the amyloid precursor protein, but it's more abundant and less prone to the aggregation and plaque formation. So here, then in addition, again, the tau protein is a protein that's involved in stabilizing microtubules in the neurons. And in Alzheimer's, it's modified and forms neurofibrillary tangles. And this occurs because it's highly phosphorylated in Alzheimer's disease, even in early stages, making it one of the earlier biomarkers. So here, quick neuron excitatory neurons were cultured, and the supernatant was collected, and the lysates were collected, and ELISA was used to measure the quantities of the amyloid beta 42, 40, and tau at the end of 1, 2, 4, and 6 weeks of culture. And here you see them graphed relative to the earliest time point of the healthy line. And they all show higher values in the Alzheimer's disease line after 6 weeks. And again, this shows that this iPS-based model system can reproduce the known phenotypes of Alzheimer's disease. So beta secretase is an enzyme that's involved in producing those amyloid beta peptides in the brain. Thus, inhibiting it results in a decrease in the amyloid beta peptides. So here, after 6 weeks, the quick neurons, excitatory neurons, were treated with beta secretase inhibitors for 3 days. And the secretion of the amyloid beta 40 and 42 in the media was then evaluated by ELISA, and it was plotted relative to the DMSO control. And an increasing dosage of the inhibitors resulted in a corresponding decrease of the amyloid beta 40 and 42. So again, this is indicating that the system can be used to study Alzheimer's as well as different pharmacological therapeutic measures. In another set of experiments using a different Alzheimer's disease line, also an ApoE4 carrier, we differentiated astrocytes using our quick glia kits and looked at the distribution of the astrocyte morphology. So apolipoprotein is highly expressed by astrocytes and is crucial for cholesterol processing in the brain. And the changes in the astrocyte morphology and the function are known to occur in neurodegenerative disorders like Alzheimer's. So the astrocytes were matured for 42 days, and then we looked at immunocytochemical detection for the GFAP, which is a common astrocyte marker. And in the Alzheimer's patient-derived lines, there was a lower GFAP, as well as more of the fibroblast-like morphology. So this shows that these cells can be used to research novel biomarkers and drugs that would impact the astrocytes, which are affected in Alzheimer's disease, but have largely been unstudied due to difficulty in accessing them. And again, here at Elixirgen, we are committed to doing our part to improve results for Alzheimer's patients. And we encourage you to visit our website where you can access more resources that we provide in honor of World Alzheimer's Month. In particular, we have many cell lines available to us, carriers of mutations, to study Alzheimer's. And in general, we have several quick tissue, again, you can buy frozen cells or kits that you can do in your own lab. We offer excitatory neurons, GABAergic neurons, dopaminergic neurons, motor neurons, cholinergic neurons, sensory neurons, as well as astrocytes, neural precursors, skeletal muscle cells, and endothelial vascular cells, if that's relevant. And in addition, we are currently working on hope to soon release microglia kits ourselves. And these, again, are highly relevant for the amazing Alzheimer's research that is currently being done.
Thank you so much, Dr. Millrod. I know we're really excited to be able to hopefully share this soon. But yeah, just as a reminder to everyone to please, if you have any questions for any of the speakers, to please use either the comment section or the questions at elixirgensci.com. We have been getting a couple of questions in there that look to be really good to be able to follow up with. Without further ado, let's move on to our next speaker, Dr. Margaret Magdesian. She received her Ph.D. in biochemistry from USP, Universidad de Sao Paulo. She is also the CEO and founder of Ananda Devices, a Montreal-based company providing faster evaluation of toxicity and efficacy of medications directly on human-derived neurons organized on a chip. Ananda technology is being used by leading pharmaceutical or biopharmaceutical companies in over 14 countries. She was also nominated for 25 awards in science and innovation, and is also an INSEAD Fellow for Entrepreneurship, and also dedicates time to supporting scientific entrepreneurship and innovation. Ladies and gentlemen, Dr. Margaret Magdesian.
Thank you very much for this kind introduction. It's a pleasure to be here and share some of the great results we are getting with your cells. And thank you especially for the previous speaker for opening the floor and really showing all the potential of those cells. And I think we have very synergistic technologies, and it will be a pleasure to share with everyone, especially at this Alzheimer's disease month, how much we can achieve with good tools in the lab. I'm sharing my screen. So Ananda Devices is based in Montreal, Canada, and what we do is we de-risk the development of therapies for neurological disorders. The company has been founded in 2015. It has been almost 8 years that we are in the market. We have a multidisciplinary team with scientists, engineers, pharmacologists, and we provide solutions of neuron-on-a-chip models to accelerate and de-risk the development of new therapies through contract research services, as well as multiple products. The favorite products in the market today are the NeuroHTS and especially the neuromuscle, and I'll talk a little bit more about those two. We have products and services that have been already validated in the market to accelerate compound screening, preclinical assays, and some of our products are even being used in phase 3 clinical trials. Our customers are mainly large pharmaceutical companies and biotechs who can really profit from our insights, especially of having a team of neuroscientists helping them. Our technology, as you mentioned, was recognized with a few awards. But what's the reason for us to be here today, and why do we do what we do? Because neuronal toxicity is the number one reason for failure of pharmaceutical products in the market. And the main issue is that products usually fail late. They fail after many years of research and after a lot of investment, because that's when they go to market, that's when they face the human. Today, animal models are still the gold standard for testing for toxicity and efficacy evaluation. However, animals do not naturally develop neurological diseases such as Parkinson, Alzheimer's, multiple sclerosis. I think animal models are great models for studying development or for studying cancer. There's a high translational rate. However, for neurological diseases, we still do not have access to the real human tissue. Besides being inefficient, slow, and costly, it's very ethically challenging to use so many animals for research and significantly challenged to scale. How can we perform the tests of so many drugs that are there? Nowadays, with artificial intelligence, we can develop so many drugs at once, but how can we validate the effect of each of those drugs? We're not the first ones to see this. The FDA, OECD, and all regulatory agencies are active trying to support companies in finding alternatives. We're one of those companies working with the FDA to offer alternatives to the market and to pharmaceutical companies. But the most important, I think what we bring really is that we can test the products in human cells at the beginning and do not leave it for the end when things are already too risky. We offer technology where we can find solutions from the start. It's also very important to set the criteria straight. So we are not replacing an animal. An animal is a very complex organism, but what we can do is we can offer a human model, a physiologically relevant model, robust, scalable, and fast. Our technology is compatible with all neuronal types. It emulates neuronal networks, and we can image 3,000 neurons in just 30 minutes. Most of the work has already been published on the technology itself. We have developed a technology that the main qualities are really speed, We can grow the neurons much faster, and scalability. So what we do is we organize the neurons on a chip, and it looks like a barcode. We can put more or less 3,000 neurons on a plate. Most importantly, this plate is compatible with standard automation equipment that is used in every lab, and we offer software to analyze the neurons very quickly. So it looks more or less like this, where we have neurons organized in standard networks. And then what we do is we check how the neurons look before treatment, after treatment. We image them very quickly. We analyze 21,000 data points, and we can correlate with the clinical imaging and genetic data from the patient. And this brings us a lot of valuable insights for the next stages of drug development. Of course, the first one is toxicity. It's very easy to detect toxicity, but there are several subtle differences in the network from one neuron to the other that we need special eyes. We need really the artificial intelligence to support us. So I'll give today an example of, just excuse me for a minute, I need to drink some water. Sorry. So I'll give here an example for some applications in Alzheimer's disease. It is Alzheimer's disease month. It's a terrible disease, and there are so many people in need for a solution, for hope. So we use cells from Elixirgen, and as you see here, those are cells from a healthy donor, from a woman, because most of the people suffering from Alzheimer's disease today are women. So these are cells from a healthy donor and from an Alzheimer's disease donor. And when we organize those cells using our technology, it's very easy to see using advanced analytics the difference in organization between the cells on the left and the cells on the right. And we see that, of course, in cells from Alzheimer's patients, they don't live as long. The axons are not that thick. They don't branch as much. And most important, most of the highlights here, the major difference, are really related to connection. All these results have been validated using RNA-seq technology, showing that indeed those are the pathways involved, or they are hyperactivated or underactivated in cells from Alzheimer's patients in relation to cells from healthy patients. The technology is very sensitive, and here I'm giving an example. This is another neuronal type, but it's basically to show how if we treat the cells with medium, they grow in a healthy network. If we add a certain compound that can be toxic, even at lower concentrations, we see a dose response difference on how neurons are degenerating. With the software, the sensitivity is even higher. Besides, we can use higher magnification and better understand what's happening in the axon itself. So as expected in neurons from healthy patients, tau distribution is quite uniform. So the networks the cytoskeleton of the cell is quite stable. However, when we look at cells from Alzheimer's disease patients, we see that the tau tangles that are in really hindering the axonal transport. And we can already predict from this data that some changes will be seen for different compounds as we test them. Everything has been validated also with biochemical assays, and we can do biochemical assays in the soma as well as in the axonal tips. And then we can use this platform, that's the beautiful part of it, to test new compounds. So basically those are cells from an Alzheimer's disease patient, and then we added a compound X. And for the naked eye, we don't see much, but if we go in detail and we test, for instance, for synapses, we see that the compound significantly increase the number of synaptic markers in those cells. So it's a promising compound in a disease where synaptic activity is decreased. Then we can go further and really dissect at least 21,000 data points per plate and quantify everything to see what's happening. Interesting, this is a compound that do not really rescue the cells, so the cell number or the number of cell death continue more or less the same. So in gray, you can see both cells are from an Alzheimer's disease patient. The red ones are treated with a drug from one of our partners. As you can see, the drug do not really rescue the cells, but it significantly increases connections, branching, axonal material, the number of synapses. So it's very easy for us to give insights to our customers on the mechanism of action and support them for faster evaluation of ways to circumvent the challenges or to test additional compounds to put together and increase cell survival. I'll give you an example. This was another customer that came to our company. They had 4 drugs. It was a biotech company. They had to decide really quickly on which of those drugs would go for animal testing. They gave us the drugs. We made a battery of tests using our technology, and we tested the drugs in cells from healthy and diseased patients, and we gave them a heat map, of course, with all the details and insights on which one would lead to the best mechanism of action, which one will be the least toxic, which one will affect the specific points that are damaged in cells from Alzheimer's disease patients. We can also keep the cells alive for very long, at least 50 days in culture, and they are still firing. So we're here basically measuring electrical function. And you can see, so healthy cells, they are beautiful. They start firing on day 14, and they keep firing over 50 days. However, Alzheimer's disease cells, they don't fire as much. So you see like day 14, the signal is still very low, but it significantly increased only after 35 days in culture. So I think this is a very important data for us to start to analyze, how those cells develop and what type of information can we extract. We use also high resolution imaging analysis, and we can get images. For instance, this is another example. Those are dopaminergic neurons. In this case was a research on Parkinson's disease, and we can see then details of what's happening in terms of axonal transport, mitochondrial activity, and different markers. As I mentioned, the technology is compatible with motor neurons, central nervous system neurons, peripheral nervous system neurons, such as motor neurons or dopaminergic neurons or excitatory neurons. And we're happy to be working with Elixirgen and have a different sort of cells that we can test in our devices. The NeurHTS is a great technology. It's a powerful tool, and we can measure at least 18 different data points and automate this measurement and put everything on a graph where we can take that a multi-parametric graph where we can take much more precise decisions and much more physiologically relevant decision in terms of which is the best drug for the right patient. Another work that we have been doing a lot, a bit out of Alzheimer's disease now, is the work with neuromuscular junction. Our company has been also involved in different 3D models. In this case, I'm showing to you, this is a model of the neuromuscular junction. So basically, it's an organoid of motor neurons connected to the muscles. It's a very efficient innervation, as you see, and it gives very precise results in response to toxins. It's so precise and it's working so well that we were capable of using this model in collaboration with UCB Pharma to bring zilucoplan to phase 3 clinical trials. So as you see, innervation is strong. We can then detect multiple toxins, including toxins or pathogenic antibodies present in the serum from patients with myasthenia gravis. Myasthenia gravis is a neurodegenerative autoimmune disease, and as you see, serum from healthy patients do not change muscle contraction, but from patient A, B, C, D, E, muscle contraction is significantly reduced. When the patients are treated with zilucoplan, the function is restored. Good tools, new tools, a lot of data. This all can support the development of new drugs, and we're happy to be here and partner with different companies for drug discovery, preclinical trials, mechanism of action assays, phase 1, phase 2, phase 3, and we really hope to be the first organoid company to bring a new drug to market. Our technology has been applied so far for Alzheimer's, Parkinson's, ALS, Charcot-Marie-Tooth, chemotherapy-induced neuropathy, myasthenia gravis, and we have given, I think, very high-quality insights in terms of compound screening, mechanism of action, neurotoxicity assays, neurodegenerative assays, neuromuscular diseases, and autoimmune diseases. It will be a pleasure working with you. If you're developing a new drug or if you need additional insights, for more information, please contact our team at contact@anandadevices.com. Thank you.
Awesome. Thank you so much, Dr. Magdesian. So on to our next portion. I guess before that, just as a reminder to everyone to please send your questions into the comments section, as well as through the email questions@elixirgensci.com. We have been getting a couple, so we'll have some pretty good questions to ask during our Q&A. But before we move forward with that, we're actually going to be introducing a new portion here where we're going to have an open discussion. So we had people send over particular topics to discuss from our experts. And I think the first one we have here pulling up is animal models versus patient-derived models. And I'll leave the floor to our two wonderful experts.
Can I start with this one?
Sure.
Yeah, as I mentioned, I think animal models have been really valuable for cancer, for developmental diseases, but animals do not develop Alzheimer's. They do not develop Parkinson naturally. What we have is models, and those models are an assumption that the disease is growing in a certain direction. Everything is based on an assumption, on top of an assumption. I think by testing with human cells, we can get a lot of insights of what's really happening in the nervous system.
Yeah. So, I mean, I agree, obviously, and, mice are not humans. And particularly with a lot of these neurodegenerative diseases, they just lack the complexity to really capture them. But additionally, IPS has so many advantages, including that it maintains genetic background, so including from disease patients. So you can study diseases that are generated by a combination of multiple genetic insults, not just, hey, let's mutate this one gene, but you're taking from actual patient samples, and therefore it's really relevant. It would be so hard to recreate that in a mouse because it's an accumulation of different factors and assaults that result in the disease. The ability to expand them and to generate multiple cell types from a particular cell line is, very unique to IPS cells, and makes them very customizable and relevant for drug testing, because you can look, you can make organoids of even different organs, not just study the brain, but does this drug also cause an arrhythmia, and look at cardiomyocytes from that same cell line, et cetera, and really develop a full model. And yet, I don't think we're entirely replacing animal models, as we've said. They do give a more holistic view. So if you want to focus and look at something that you kind of are expecting, you can create an IPS-based model and look at that cell type. But if you want to see, maybe capture some unexpected results, that behavioral changes that you wouldn't necessarily see in that kind of context, still does play a role.
Okay, so I mean, I know I've had a lot of people that I've talked to during my research times, and even now as well, that there can be where they want to stick more with the animal models because they can get more of a in vivo model type of approach. Do you think that the patient-derived iPSCs could help with that for that next step, or even just any other potential models?
I mean, I think they've definitely made progress. We now have, microfluidics it's not just a 2D culture. We're providing fluidics. There are organoid models that take into account, structures and biomechanics and stuff like that. So it's definitely more advanced than it has been.
Yeah, I think we have to take into account, just with the cells, the HeLa cells, how much progress has been made in medicine, right? So the cure for AIDS was found in a lot of research on HeLa cells. A lot of animals were used as well, but HeLa cells brought so many insights into the union. I think we are in a transition phase. It will be really, more and more of those human models will come up. they will be better and better organized, as much closer to what's really happening in humans in terms of 3D organizations, network organizations, innervation of tissues. And from there on, we'll be slowly transitioning to what really is more physiologically relevant, and ideally move all the way to precision medicine, where we'll be able to understand how each patient responds to each medication.
I mean, and again, traditionally, some of the challenges of iPSC-based models was, that it required people skilled in culturing iPSC, which can be finicky. That generating the lines could take a while, although I would argue less time than generating some of these highly, customized mouse models. But with companies like Elixirgen, those obstacles can be surmounted because you can, if you're interested in studying neurons, you don't really need to deal with iPSC. You can order the neurons from us, or and work with us to see which particular lines you're interested in, what mutations, things like that. So it's now at a point that it's much more usable for the neural and applied research.
And if you don't have a lab, you can even contact us and we'll give you just the report if your drug works or not, if it's toxic or not.
Right. Yeah.
No, that's definitely valuable. So yeah, I think on that, or on those points, maybe actually I think that kind of carries over into our other discussion topic as well. Where I know a lot of questions that we definitely get quite a bit is the variability across these patient lines. So now moving from animal model versus patient-derived, like now getting to the specifics of these lines, there can definitely be high variability. So how do the both of you feel this issue could either be remedied, or what are the common issues that pop up with this variability?
So I think, first of all, the variability in iPSC, can come from different sources. It can come from, interdonor, meaning difference between different patients. It can also come from different clones within the same donor, or different batches, et cetera. And that's been looked at extensively, and we found that most of the variability really comes from interdonor variability, the difference between different donors. And yet that's real. Like, everybody is different, and you want to make sure that your drug is not going to work just on a hypothetical average person, but on a full gamut of the population. And so the way to surmount that, just like any clinical study, is just to have a lot of cell lines, and you need to increase the statistical power by working on more cell lines, more independent donors, et cetera.
I think this is a plus. I think this is tremendously important, because today in the US, I think I don't have the numbers on top of my head, but one of the key problems is the side effects of medications, because also the medications don't fit every patient. So having the opportunity now to address the whole variability and testing different cell types of the same medication with different cell types, this is amazing. And I'll give an example. In this myasthenia gravis test work that we were doing with UCB Pharma, for instance, some patients, they have an autoimmune antibody that block a certain activity in the neuromuscular junction. Those patients don't respond to one of the drugs, and we can detect this with our device, exactly who are the right patients for the right medication. And this is all being enabled by the use of individual cells from individual patients, or by the use of samples from individual patients. I think we have to look at this as how can we really address the best therapy for the patient. And yes, variability in biology is always around 20%. I think, I've been in this, doing cell culture for so long to be sure that this is almost inescapable. But the beautiful part of it is, like, when we have a significant compound or a significant disease like Alzheimer's, you see, like, it's not a 20% variability, like it's no connection, a very low connection number. If a drug has an effect that can increase by 30% the function in patients, I would take it, right? And that's how the new compounds are coming out. Or spinal cord injury, if you can make 5% of the neurons regrow and eventually help function, of course, I think that's we should try.
Yeah. I would also just like to add another advantage of some of these iPSC approaches that's relevant to variability is that you can actually use like targeted nucleases or different gene editing tools to make isogenic controls where the whole cell line will only differ by the presence or absence of whatever genetic variant you're interested in studying. And that's obviously easier to do with cell lines than with mice. And that allows you, you should be studying multiple cell lines with multiple genetic backgrounds, et cetera. But for each one, you can also make these controls to really tease out that the effect that you're seeing is from what you think it is.
I think. Were there any other points either of you wanted to bring up a little bit more in regards to variability, or do we want to potentially move over into our Q&A, which I can tell you we have gotten quite a bit of engagement from our attendees.
Let's go to the Q&A.
Sounds like a plan. All right. So I'll go through. So I think one of our first questions we have here is for Dr. Millrod. I think, yeah. So this is one that we received through our email. For slide 12, the question is, have you done this ELISA assay for other compounds?
So we've done this assay for different customers with compounds that they manufacture that they're interested in, and compared it to these, which were acted as our positive controls, basically. That's one of our standard assays that we offer as a service. And again, just, contact us through any of the various channels we've mentioned, and we're happy to work with you on designing what would help you.
Perfect. And then I think just a quick follow-up, since I do see that was another question in the chat. On the next slide 13, the question was, what kind of p-tau was measured in the supernatant, as well as, or how about oligomeric tau as well?
Yes, so this was looking at phosphorylated tau 181. We did not look at the oligometric tau.
Gotcha. All right. Okay, let's. I think we have some questions for you as well, Margaret. I think the first question we got through the email was, does the technology at Ananda Devices apply only to Alzheimer's cells, or are there other models?
Yes, no, we have tried it with multiple diseases. Like the beauty is like the technology is compatible with all neuronal types. So we have already applications in Alzheimer's, Parkinson, multiple sclerosis, and also with myasthenia gravis and neuromuscular diseases as well.
I think we have some others in the chat as well. So we have another question for you, Dr. Magdesian. Do you help establish these setups in the individual labs?
Yes, we can do that as well. So we offer services. If a company would like, they just send us the compound, we test, and we give them the report. But we also offer training services. If they would like to use our technology, we train the team. And in some cases, the companies can acquire the plates and perform the assays themselves.
Then I think an additional follow-up for yours as well is your Neuro HTS microplate containing a sensor to measure electroconductivity? As well, does this work with cerebral organoids, or have you tried?
Yes, this is the work that we do as a service. We have fully equipped labs, and we perform the analysis using our technology on top of MEA plates, and we get all the results.
And then I think I have another question over to Dr. Millrod. So for back to slide 12, the question is that only from excitatory neurons? Have other neuronal lines been tested?
So this particular experiment we performed with our excitatory neurons. There are obviously other cell types that are highly relevant to Alzheimer's disease that should be looked at. We have not done that yet. But I don't see a reason why it wouldn't, someone couldn't try. We would be happy to try with you if you reject us.
Sounds good. All right. As well, I guess over to slide 18, there's another one written in that asked, what experiments have been done on the microglia cells?
So again, these are still under development. They're not fully released, but as you can see here, we did a phagocytosis assay showing that the microglia are functional. And we looked at immunochemistry as well as PCR, looking at for known markers. But we haven't used them in any, complex experiments or looking at Alzheimer's markers or things like that yet, because the cell type is still in its infancy of development.
But once we have that developed, we will probably be moving towards that, right?
Right. Yeah.
Perfect. And then, so I'm not sure specifically. I think this question might be for you, Dr. Magdesian. Do you have an in vitro blood-brain barrier model available through Ananda?
Not yet, but we are evaluating a very nice one, and we should have news soon.
Right. And then I think we have another question in the comment section for you, Dr. Millrod. So I'm not going to read through all of it because I know that there are a couple of things that are looped in on there. But I think the question is kind of focusing towards how the viability of neurons are after freezing, not only just the neurons, but astrocytes and potentially pericytes as well.
Yeah. So, I mean, we do look at that as part of our quality testing and assurance. So when we freeze cells, we freeze enough in a vial that when you thaw, you will have enough to use for your experiment, that, the amount that we guarantee for that particular cell type. Typically, we have more than 50% of the cells survive. But again, we take that all into account. So if you're buying 5 million cells, we will give you a vial that will give you 5 million live thawed cells. And we test each lot and verify it before we send it out.
Good. I do have another question here for Dr. Magdesian. So they saw that you're able to measure firing frequency. Is there a possibility to look at single cell activity? For example, perhaps an option to temporarily access the somas for whole cell patch count?
Yes, we have an assay specifically for that. This is in our, as I mentioned, we have been in the market for 8 years. We have developed a lot of assays, and usually when the customer comes to us, we review with them a large variety. So here we are presenting just a little bit of what we can do, but please reach out to us. It will be a pleasure to share some of the beautiful images we have.
And then I think for Dr. Millrod, there is a question in regards to access to early onset Alzheimer's disease lines.
So again, we have access, easy access to all of the CERM lines. I'm not sure if they have an early onset Alzheimer based line, but they very well might. Again, if you go to our website and the Alzheimer's page, which will list some of those relevant cell lines. I haven't specifically looked and checked if that includes that, but also if there's a cell line that you know of, or that we can put you in touch with also companies that can generate it for you. So we can facilitate procurement really of any cell line of interest, and then use our differentiation protocols on it to get you what you want and need.
Yeah, so that would also include being able to work with groups with help generating of isogenic controls, correct?
Yeah, right.
Perfect.
Yeah, we don't generate the iPSC lines here, but we can help obtain them or connect you to companies that do that. And it could be that they do already exist, and if CERM has them, then it's even easier. But yeah.
Perfect. Right. And then I'm just checking on. I think we have I think we have potentially, oh yeah, we have one more question through the email chain. So can Elixirgen and Ananda help with AD grant application support?
So yeah, we've definitely helped people with grant applications and provided, any documentation that they need to support that. We've done that for several customers, so we are definitely familiar with the process.
Yes, in the US, in Canada, and in Europe as well.
And right, I think we might have time for one or two more questions. Let's see. For you, Margaret, does Ananda Devices only have 2D cell culture models? That was another question through the questions at.
We have we call it 2.5, the neuro HTS, because the cells are organized in precise networks. We have the 2D, 2.5, and also the 3D models. The neuromuscular junction on the chip is a 3D model.
All right. And then I think a new question just came in through the chat. Or can the iPSC passage neurons to keep? Or sorry, I need to reread that. I apologize. Yeah. All right, I will work on this question real quick. I do apologize. I'll bring in another, or I think we have one other question from the email section. For you, Margaret, with the growing landscape for MPS devices, what differentiates Ananda's organs on a chip model from others?
Well, it's easy. It's speed and scalability. So first, we offer a technology that the data can be acquired in 3 weeks, 2 to 3 weeks. We deliver a lot of data of the neuronal organization, network formation, survival, potential mechanism of action. And the second big advantage is really scalability. We can test multiple compounds at once and compare, or we can test cells from multiple patients at once and compare the results, guiding our customers in the best application for their compound.
And then, thank you again for who clarified the question that I couldn't read. I apologize. For Dr. Millrod, can the cells be passaged and frozen? The differentiated ones that we showed.
So it depends on the cell type. Our kits give instructions, and when you buy the thawed cells, typically we do passage them. Once they cannot be expanded indefinitely because they're not iPSC anymore, which is a good thing. They are terminally differentiated, so there's a limit. Some cell types we give with instructions on how to freeze and make stocks that you could have for expansion a little bit, but most of them are terminally differentiated. I mean, they're all terminally differentiated. They're not iPSC, so they don't expand forever. But you can passage and keep them going in culture for a while and expand them.
Now, one of our technologies from a previous webinar, we were able to have, in particular, I think our excitatory neurons were able to grow over 10 plus weeks, right?
Yeah, we've done MEA assays on them after an extended period of time, after we ship them across, internationally, things like that. So they are fairly durable.
Even we used them for 50 days. It was firing and healthy and everything as well.
And then I think we have one more question. I think we can fit it in right within time. This one is for you, Margaret. The question is your plate or device with software? Which one is really used for this particular model?
The software is so the companies can take images, analyze, and send it to us, and we will deliver the report.
Perfect. All right, well, if anyone else has questions, please shoot them over either in the comments or through the questions at elixirgensci.com, and we'll be able to make sure to reach out to you to follow up. But I wanted to say thank you very much to Dr. Millrod and to Dr. Magdesian for giving this wonderful talk and discussion. Also, to let everyone here know, we have an upcoming event. We will be at Neuroscience 2023 SFM this year. We will not only have a booth at 238, but we also have a nice poster and there's some pretty exciting data to share on that one. So please swing on by, say hi, and get to chat with me and a bunch of wonderful experts here at Elixirgen. Thank you all again. And as just a reminder, this webinar is really focused on the Alzheimer's Awareness Month. And, you know, for next year, we're planning to have this again. So if you'd like to contribute to any of these resources and events that we've been hosting through, please contact us at info@elixirgensci.com. Once again, thank you all for taking the time to join us today. And just as a reminder, everyone who registered will have access to the recording of this webinar. And yes, thank you again to our wonderful speakers, and I hope everyone has a wonderful rest of your day and weekend. Bye, everybody.